To explain about genomic and cDNA libraries. First, what is a genetic library? Gene libraries or DNA libraries are collection of cloned genes that are big enough to contain at least one copy of every gene from a particular organism. These libraries are of two types: genomic library and cDNA library. First, genomic library. A genomic library is a collection of the total genomic DNA from a single organism. The DNA is stored in a population of identical vectors, each containing a different insert of DNA. It is created using molecular cloning, and the genome size is expressed in terms of base pairs. In prokaryotes, the structural genes coding for proteins are continuous, while in eukaryotes, the coding regions exons are separated by non-coding regions introns. Therefore, the construction of gene libraries for eukaryotes is more complicated. And the steps for constructing a genomic library: isolation of DNA from cells, digestion into small fragments, introduction into suitable vectors, insertion into bacteria, DNA isolation, and the last step is collection of genomic DNA library. Isolation of DNA. In eukaryotes, prepare the cell nuclei, remove proteins, lipids, and other unwanted macromolecules by protease digestion and phase extraction. In prokaryotes, they extracted DNA directly from the cells. Digestion into small fragments. Physical shearing. It can be done by two types: pipetting and mixing. Restriction enzyme digestion. Partial digestion is preferred to get greater lengths of DNA fragments. Introduction into suitable vectors. Each fragment is different and have a unique DNA sequence. Inserted into suitable vectors including plasmids and bacteriophage vectors. Vectors are digested with the same restriction enzyme and sealed to human DNA using DNA ligase enzyme. The resulting molecules are recombinants. Insertion into bacteria. Inserted into host bacteria, which is mostly E. coli. The microbes are grown in culture. They are made to take up the DNA. They replicate their genome along with the vector genome contained within them and produce clones of the original genome. The collection of clones, which contains all the sequence found in the original source, including the sequence of interest, forms the genomic library. Multiplication and production of clones, independent plasmid replication and host cell replication. Screening of clones. The methods used for the selection of recombinants include expression screening or direct selection of recombinants, insertional selection inactivation method, blue-white selection method, colony hybridization technique. Uses of genomic library. Researchers can explore the genome of an organism to learn more about genomic structure and function. They can map the genome, identifying the locations of specific genes. It is useful in recombinant DNA technology. It is the first step in any DNA sequencing projects. It helps in the identification of novel pharmaceutically important genes. Next, the cDNA library. cDNA. Complementary DNA is produced from a fully transcribed messenger ribonucleic acid, that is messenger RNA, that contains only the expressed gene of an organism. Clones of such DNA copies of mRNA are called as cDNA clones. cDNA library. A cDNA library is a combination of cloned cDNA fragments constituting some portion of the transcriptome of an organism, which are inserted into many host cells. Isolation of mRNA. mRNA is first isolated from a cell type or tissue of interest. It may be desirable to remove highly abundant tRNAs and rRNAs, which might otherwise constitute the majority of the final library, to the determinant of the detection of low abundance RNAs. Synthesis of the first strand of DNA. As mRNA is single stranded, it is first converted into DNA to be introduced into a suitable vector. A short oligoprimer is annihilated to the poly A tail of the mRNA. Reverse transcriptase extends the 3' end of the primer by mRNA molecule as a template, producing a cDNA mRNA hybrid. The mRNA from the cDNA mRNA hybrid can be removed by alkaline hydrolysis or RNase H to give a single stranded cDNA molecule. The second strand of cDNA generation. The single stranded cDNA is converted to double stranded cDNA by either RTase or E. coli DNA polymerase. Incorporation of cDNA into a vector. Double-stranded cDNA can be trimmed with S1 nuclease to obtain blunt-ended double-stranded cDNA molecule, followed by addition of terminal transferase to tail the cDNA with CS and ligation into a vector. Because this blunt-end ligation is inefficient, short restriction site linkers are first ligated to both ends. Cloning of cDNAs. cDNAs are commonly cloned in phage insertion vectors. The bacteriophages are more desirable when a large number of recombinants are required for cloning low abundant mRNAs, as recombinant phages are produced by in vitro packaging. It can be easily handled and stored when compared to bacterial colonies carrying plasmids. Thank you.